So let's take a look at this diagram here so that you can see what I'm talking about. On the y-axis in your diagrams, you have the free energy. So the free energy could be increasing or decreasing. And on the x-axis, we have the process of the reaction. So as the reaction is going forward from left to right, you can see that the energy level initially increases in this reaction and then decreases dramatically. So the substrates start out here. They have that much free energy stored in their chemical bonds. The products have less free energy stored in their chemical bonds. So the net change, which is this much energy, is negative because you have lost energy to the environment. However, initially, there had to be an input of energy before the reaction could happen. This input of energy is called the activation energy. This is going to bring the substrates to the transitional state, and at the transitional state, the high energy, the additional energy added to the chemical bonds makes them unstable enough that they can be broken and reformed, and then the reaction can go forward. If you don't reach the transitional state, the reaction does not go forward. So you have to provide enough energy to the system to get to this state before anything will happen. In the second diagram, here are your substrates, here are your products. There is your change in free energy, positive or negative? Positive. So this is endergonic, which is non-spontaneous. And this still requires that initial input of energy, the activation energy, for the substrates to reach the transitional state before the bonds could be broken and reformed. If you don't reach the transitional state, you cannot break and reform those bonds. The reaction will not go forward. So regardless of whether it's endergonic on the right or exergonic on the left, both of them require an activation energy. And different reactions require a different amount of activation energy. That activation energy usually comes from the heat in the environment. That makes those molecules vibrate more, and if they have absorbed enough heat from the environment, they can reach this transitional state, and then the reaction can go forward. Does that make sense to everyone? Yeah. Any questions? All right. So if I were to give you diagrams like this without actually drawing the arrows for you, could you figure out the activation energy on the diagram as opposed to the delta G on the diagram? Great. That's about as much as we want you to be able to do at this level. Right. But we are more interested in what happens in reactions inside of the cell when you have these endergonic reactions that require energy. How do you carry those out? Many of the reactions in your cell require energy. So we're going back to energy coupling. Just as you saw with cotransport, the movement of protons down their gradient released energy. The movement of sugar against this gradient required energy. You couple the two of them together in cotransport. You can do the same thing with reactions. You have an exergonic reaction, you couple it with an endergonic reaction. Release of energy from one used to carry out the other one that requires energy. And usually, that energy is coming from hydrolysis of ATP, which is a highly exergonic reaction. So, remember ATP, 3 phosphate molecules here. These are relatively unstable bonds. And if you were to remove the first phosphate here, the diphosphate is more stable than the triphosphate. So going from a relatively unstable ATP with 3 phosphate groups, 3 negative charges all repelling each other, to something that only has 2 phosphate groups, only 2 negative charges repelling each other, that's going from something highly unstable to something more stable. That reaction goes forward spontaneously, and it will release energy. So when you remove one of the phosphate groups, that releases energy. How much? About 7 kilocalories. So this hydrolysis of ATP, removing one of the phosphate groups to make it diphosphate, releases approximately 7.3 kilocalories of energy per mole. Which is good because you can then use that energy to drive other reactions that require energy if you couple them together. So let's take a look at an example. Glutamic acid and glutamine are 2 different amino acids that your body uses to make proteins. And to make glutamine, you need to add an ammonia to glutamate, which is glutamic acid. So you end up with an amino group here. So there's an additional amino group on the amino acid. And that reaction is endergonic. It requires 3.4 kilocalories per mole, because you're taking two small molecules and joining them together. So you're decreasing the entropy of the system by making a larger molecule. And that requires energy. ATP hydrolysis releases energy, because you're taking one big thing and breaking it into smaller things. So you're going from something relatively unstable to something more stable, and that's going to release some energy. It's negative 7.3 kilocalories, which is more energy than you need to make glutamine. Glutamine synthesis by itself is non-spontaneous, endergonic. But if you couple the two of them together, then you end up with a reaction that is overall exergonic. So when you hydrolyze ATP and stick the phosphate on glutamic acid, and then glutamic acid and the phosphate attached to it is reacted with ammonia to make glutamine, the overall reaction has a delta G of negative 3.9 kilocalories. So you went from a biosynthetic pathway that was endergonic to an overall biosynthetic pathway that is now exergonic. So what happens to the extra 3.9 kilocalories per mole that is being released from ATP hydrolysis? Release as heat. Heats up your cells. So far, so good? Okey doke. ATP hydrolysis is not just used for chemical pathways like that. Sometimes you need motion of certain molecules in your cells that requires energy, and we're going to take a look at the role of helicase, which is this enzyme, in DNA replication towards the end of the semester. Helicase moves along the length of the DNA, and it's going to break the hydrogen bonds that hold the two strands together, and that requires energy. So this movement, the changing of the shape of helicase, requires ATP. So this is not a chemical reaction that is going to be endergonic or exergonic. It's the movement of helicase that requires energy. So we'll look at that later this semester. All right, do you know what a catalyst is? I've asked you this question at the beginning of the semester. So what is a catalyst? Yes, sir. Increases the rate of the reaction and what happens to it? Yes, sir? Does it get used up? The point of the catalyst is that it doesn't get used up. So it can be recycled over and over and over again. So it does reduce the activation energy in the case of enzymatic reactions. It is something that increases the rate of reaction, but itself is not affected by the reaction. So that's an important point to catalyst. So the catalyst is not affected by the reaction. It can be used over and over and over again. And enzymes are catalysts, and they're usually proteins, and you know of exceptions to that. What's the famous exception you know of? RNA. Yes, RNA called ribozymes, yes. So ribozymes are enzymes that are made up of RNA. So most enzymes that you deal with are proteins, but there are exceptions like the ribozymes. So we're going to look at a simple example. Sucrase is the enzyme that will break down sucrose. So the ASE ending is very typical of enzymes. So usually when you see a protein name that ends in ASE, you think enzyme. So sucrase breaks down sucrose. So here's sucrose. It's a disaccharide. It has a glucose and a fructose joined together. And it's a hydrolysis reaction, so you're adding a water molecule to break this covalent bond here. And so you're going to reform the glucose and the fructose. Would you expect this to be spontaneous or non-spontaneous? Spontaneous, why? You're taking a bigger, more complicated molecule and breaking it into smaller things, so you're increasing the entropy of the universe by doing that. And so that's usually spontaneous. Now, sucrose is table sugar, so if you leave your table sugar out on the table, is it going to break down into glucose and fructose? Theoretically, yes. Practically, it's going to stay as sucrose for a very long time. Why is that? You could dissolve it in water and see what happens. Will it break down into glucose and sucrose in water? Why not? It is spontaneous. Yes, sir. Well, why do you need the enzyme? It is spontaneous. What do you need before the reaction goes forward? You need that activation energy. So the breakdown of sucrose has a high activation energy. It's not going to happen very often. If you just put it in, you can dissolve your sucrose in water and let it sit there for a long time, and it's just not going to go forward because those sucrose molecules just will not reach the transitional state because they need a whole lot of activation energy to get up there. If they do get up there, then the reaction will go forward spontaneously. But it's just the trick of getting them to this transitional state. It requires a lot of energy from the environment for that to happen. So the enzyme's role in your cells is to help this reaction go forward by lowering the activation energy. So this diagram is showing you the difference between an enzymatic reaction that has the enzyme present and doesn't have the enzyme present. Same reaction. So you're starting with the same substrates, A and B, C. At the transitional state, they're going to be very momentarily joined before the bonds break and reform, and then you get your products. But you have to reach the transitional state. Without the enzyme, you need a lot of energy to reach the transitional state. The only thing the enzyme does is lower the activation energy level. So the transitional state does not require that much energy for the substrate to get to. So you only need a little bit of energy from the environment to get to this state. So in the presence of the enzyme, you do not need to heat up your cells too much for this reaction to go forward. In the absence of the enzyme, sucrose is just not going to break down that easily. Even though this is spontaneous. But you have to get to the transitional state first before the reaction goes forward. You add sucrase to the solution, it happens much more quickly. Because now you don't need that much heat from the environment for this to happen. Room temperature is good enough. So far, so good? All right, one question. What does the enzyme do to the delta G for the reaction? Is there a difference between the delta G here and the delta G there? No. The enzyme does nothing to the delta G. It only affects the activation energy. So I'm going to show you two more diagrams that basically go over the same thing. One is for an exergonic reaction. Enzyme lowers the activation energy, does not affect the delta G. And here's one for an endergonic reaction. Enzyme lowers the activation energy, does not affect the delta G. So delta G is not affected by the enzymes at all because your substrates and products haven't changed. It's the same substrate, same product. The only thing that matters is that the enzymes lower the activation energy, so more and more of the substrates can actually reach the energy level of the transitional state. There is enough heat in the environment to allow them to reach this, but very few of them will actually absorb enough heat to get to the transitional state in the absence of the enzyme. So the reaction will be very slow without the enzyme, but a lot faster with the enzyme. So how do enzymes actually do this? So remember enzymes as proteins, three-dimensional shape. They could have tertiary structure or quaternary structure if they are made up of multiple polypeptides. And they have what we call an active site, a specific region in the protein, the shape of the protein where the substrates can bind. And then the substrate binds there, it actually causes what we call an induced fit. So the protein shape changes around the substrates, and then as the reaction goes forward and the products are released, the enzyme goes back to its original.